Okay, so in today's class, we are going to discuss the last step of the aerobic respiration, that is electron transport system, which is also called as ETS. It is also called as electron transport chain, or it is also called as terminal oxidation because the oxidation is taking at the end of the end stages. So we can also call this as terminal oxidation. It is also called as oxidative phosphorylation, since there is formation of ATP, and by oxidation process we also call it as oxidative phosphorylation. So the electron transport system, so you can notice that it is a metabolic pathway which is present in the inner mitochondrial membrane. So the cristae will be having the Rakus particles in which this electron transport system takes place. So electron transport system is the metabolic pathway present in the inner mitochondrial membrane through which electron passes from one carrier to another. So why do they have to pass from one carrier to another? As they release and utilize, this is to release and utilize the energy stored in NADH2 and FADH2, which is formed during TCA cycle. So they are energy rich molecule. So when the electrons are flowing, this also facilitates the release and uh, utilization of energy which is stored in NADH2. See, one NADH2 is equivalent to 3 ATP and one FADH2 is equivalent to 2 ATP. So these NADH2 and FADH2 are formed during the Krebs cycle or tricarboxylic acid cycle. So that energy rich molecules, the energy has to be released and utilized. That happens during this electron transport system, and it takes place by oxidation. So the electron transport system is to release and utilize energy stored in the energy rich molecules like NADH2 and FADH2, which are formed during TCA cycle by oxidation process. So the electrons are passed on to oxygen to form water. Finally, the electrons are passed on to oxygen to form water. So electrons from NADH are oxidized by an NADH dehydrogenase. We call it as complex 1. So complex 1 is an enzyme system. Okay. So electrons are then transferred to ubiquinone Q, which is located within the inner membrane. So electrons from NADH are oxidized to uh, by an NADH dehydrogenase, and we call it as complex 1. So you can notice here. So the NADH is oxidized by an NADH dehydrogenase, and this enzyme system we call it as complex 1. This is taking place within the inner mitochondrial membrane. Okay, electrons are then transferred to ubiquinone. So you can notice that from here the electrons are passed through the carriers through the to the ubiquinone located within the inner membrane. Okay. Ubiquinone also receives reducing equivalents via through the FADH2 complex. So they also receive the uh, equivalents via FADH2, that is complex 2. So they are going to receive the electrons from the ubiquinone is also going to receive the electrons from the FADH2 complex 2, that is generated during the oxidation of succinate in citric acid cycle. So this is generated during the oxidation of FADH2, which is generated during the oxidation of succinate in citric acid cycle. So that FADH2 is going to also reduces at complex 2 via FADH2. So the enzyme involved is succinate dehydrogenase. The reduced ubiquinone, from the reduced ubiquinone or QH2. So the reduced ubiquinone or QH2 is then oxidized with the transfer of electrons to cytochrome C. So they are oxidized through the electrons to with the transfer of electrons to cytochrome C via cytochrome BC1 complex. So this we call it as complex 3. So there is complex 1, enzyme system, that is NADH dehydrogenase, where NADH is reduced, and there is complex 2 where the FADH2 is reduced when the electrons from the ubiquinone passes through this. So the FADH2, which is a product obtained during the uh, oxidation of succinate. So the succinate dehydrogenase. So that takes place in the complex 2. In the complex 3, the reduced ubiquinone or ubiquinol or QH2 is then oxidized with the transfer of electrons to cytochrome C. So the electrons are transferred to cytochrome C via cytochrome BC1 complex. So this we also call it as complex 3. The cytochrome C is a small protein. You can notice here the cytochrome C is a small protein attached to the outer surface of the inner membrane. It acts as a mobile carrier of electrons between complex 3 and complex 4. So it acts as a mobile carrier of electrons between complex 3 and complex 4. So that is what they act as. So they act as a cytochrome C. You can notice here the cytochrome C is a small protein attached to the outer surface of the inner membrane, and it acts as a mobile carrier of electrons between cytochrome between complex 3 and complex 4. So they are going to provide the electrons to the complex 3 and complex 4. So they are going to supply the electrons from complex 3 to complex 4. So the complex 4 refers to the cytochrome uh, C oxidase. So what does this complex 3 refer 4 refers to complex 4 refers to cytochrome C oxidase. So these complex systems are a system of enzymes that they have. So complex 4 refers to cytochrome C oxidase. So this complex contains cytochromes A1, A3, and two copper centers. You can notice this copper centers. So Cu2. So two copper centers. So they have complex 4 refers to cytochrome C oxidase complex, which contains cytochrome A and A3, and they have two copper centers. Okay. So that is the complex 4. So when the electrons pass from one carrier to another via complex 1 to complex 4, they are coupled to ATP synthase. So they are coupled to ATP synthase, and there is the formation of ATP synthase is the complex 5. So they are coupled to ATP synthase for the ATP production. So they are coupled to ATP synthase, which is the complex 5 for the ATP production. So the number of ATP molecules produced depends on the nature of the electron donor. So the number of ATP molecules produced depends on the nature of the electron donor. If oxidation of one NADH takes place, the energy rich molecule NADH2, when they undergo oxidation, they are going to form three ATPs. And oxidation of one FADH2, you already know what is NADH, okay, nicotinamide adenosine dinucleotide. So if it is having H, we call it as reduced nicotinamide adenosine dinucleotide. So they are going to produce 3 ATP. Flavin adenosine dinucleotide. So that is reduced flavin adenosine dinucleotide. So they are going to undergo oxidation to produce 2 ATP. So here you have got a list of enzymes that are there. So initially the NADH is going to get oxidized by the complex one, that is NADH dehydrogenase. FADH2 is oxidized by the complex 2 FADH2, that is succinate dehydrogenase. Complex 3, they are having the cytochrome BC1 uh, complex. Complex 4, they have the cytochrome C oxidase, and complex 5, they have this ATP synthase. So these are the complexes that we have. So cytochrome C oxidase is present in complex 4. So complex 3 will be having the cytochrome BC1 complex, and complex 2, they will be having the FADH2. So NADH dehydrogenase, complex 2 is FADH2, complex 3 is cytochrome BC1, complex 4 is cytochrome C oxidase, complex 5 is ATP synthase. So that is how the energy rich molecules are reduced to 3 ATPs, like NADH2 is reduced to 3 ATPs, and from one FADH2 we obtain 2 ATP. So overall electron transport system, you can write it in the simple part of it. So NADH plus H plus is reduced, it is oxidized in the complex one, that is NADH dehydrogenase. So the electrons are carried by the quinone to the complex 2 FADH2, where the FADH2 is obtained from succinate. Okay, the complex 3, the cytochrome BC1, so the electrons are passed from this cytochrome C carries this electron to cytochrome 4, that is cytochrome C oxidase, where there's a uh, finally and it carries to water. So the overall electron transport chain is this. They have 5 complex systems. So the details of the enzymes and the carriers we have here. Okay, complex 1 consists of NADH dehydrogenase. Complex 2 consists of FADH2. Complex 3 consists of cytochrome BC1. Complex 4 consists of cytochrome C oxidase. Complex 5 ATP synthase. See, in aerobic respiration, the role of oxygen is limited to the final stage, that is the electron transport system. Yet, oxygen is vital since it drives the whole process by removing hydrogen from the system. So, oxidation is an important process since it drives the entire process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor. So you should understand that oxidation acts as the final hydrogen acceptor. In respiration, energy of oxidation reduction is utilized for the phosphorylation, that is synthesis of ATP. Okay, so oxygen acts as the final hydrogen acceptor to form water. In respiration, energy of oxidation reduction is utilized for the phosphorylation, that is synthesis of ATP. So this process is called as oxidative phosphorylation. So this process, we call it as oxidative phosphorylation. See, the oxidative phosphorylation is different from photophosphorylation. So the oxidative phosphorylation is different from photophosphorylation. Here, light energy is utilized for the production of proton gradient for phosphorylation. So photophosphorylation, it takes place in photosynthesis and they require light and they take place during the light reaction. Okay, so we call it as photophosphorylation. Here, it is taking place in photosynthesis and light energy is utilized for the production of proton gradient for phosphorylation. But in case of respiration, oxidation is utilized for the production of proton gradient for phosphorylation. So that is the difference between oxidative phosphorylation and photophosphorylation. The energy which is released during electron transport system is utilized to synthesize ATP by ATP synthase, that is in the complex 5. ATP synthase has 2 major components. So we have already discussed about that. So the F0 integral membrane, if you can recollect this chemiosmotic hypothesis. So I would have discussed about this role of ATP synthesis. It has 2 major components. F0, which is the integral membrane protein complex, and F1 head piece, which is the peripheral membrane protein complex. So the energy released by an electron transport system is utilized to synthesize ATP by ATP synthase, which is complex 5. ATP synthase has 2 components, the F0, which is the integral membrane protein complex, and F1, which is the headpiece. F1 is the headpiece, which is the peripheral membrane protein complex. So the F1, the peripheral membrane protein complex, is the site for ATP synthesis from ADP and inorganic phosphate. So this is the site. F1 is the site for the synthesis of ATP synthesis is. taking place in the F1 complex or F1 headpiece from ADP and inorganic phosphate, they are synthesizing ATP. So F naught is the integral membrane protein complex. It forms a channel through which protons cross the inner membrane. So it forms a channel through which the protons cross the inner membrane, and this proton crossing through the inner membrane is required for the activation of ATP synthase. Only then the ATP is synthesized. We have already discussed this concept in chemiosmotic hypothesis. So the F naught integral membrane protein complex, it forms a channel through which protons cross the inner membrane. So it is forming a channel through which protons cross the inner membrane. Okay. So only when they are protons are crossing through the inner membrane, ATP synthase is activated. So the passage of protons is coupled to the catalytic site of the F1 component for ATP production. For each ATP produced, two protons passes through F naught. So for each ATP produced, two protons passes through F naught from the intermembrane space to the matrix down the electrochemical proton gradient. So for every ATP produced, two protons passes through this F naught from the intermembrane space. So from the intermembrane space to the matrix down the uh, electrochemical photogradient. That is what is happening from the intermembrane uh, matrix space to the matrix down. See, you can notice this intermembrane space. From the intermembrane space down the electrochemical proton gradient into the matrix. From the intermembrane space to the matrix down the electrochemical proton gradient. So you can notice that in between the outer and inner mitochondrial membrane, there is this inner membrane, intermembrane space. So for each ATP that is being produced, so the protons, two protons passes through the F naught from the intermembrane space to the matrix down the electrochemical proton gradient. So you can understand by this picture. Okay, so this is the outer membrane, this is the inner membrane. So the intermembrane is present between the outer and inner membrane. There is the intermembrane space. So the protons from the intermembrane space passes through F naught from the intermembrane space to the matrix down the electrochemical proton gradient. So this is what you can remember about. So you can see here, this is the diagram that has been given in your book. So the cytosol, so the outer mitochondrial membrane, this is the cytoplasm. Cytosol implies cytoplasm, the outer mitochondrial membrane, inner mitochondrial membrane. In between them, there is intermembrane space, which has the protons. So you can notice that the complex system 1, 2, 3, and 4, they are all located in this uh, inner membrane, and complex 5, which has this ATP synthase. Okay. So the matrix, you can notice here, the complex one has NADH dehydrogenase. And when the protons are passing through them, so the NADH2 is reduced to NAD plus, NAD plus, and the protons is passed to the intermembrane space. And from this complex one, the electrons are carried to complex 3. See, this is a very good diagram which describes it. From complex 1, the electrons are carried to complex 3. So where uh, they are further carried to complex 4. So the, and from the complex 2 also, the electrons are carried to complex 3, where it is FADH2. FADH2 is reduced to FADH. FAD, that is, it is oxidized, and the electrons from this complex 2 also reaches the complex 3. So the electrons received from complex 1 and complex 2 is carried from complex 3 to complex 4. So you can notice here, and from the complex 4, so where the oxygen is adding to H2, H plus 2H plus to form water. Okay, so from this complex 4, finally the Now, once these are oxidized, okay, now the complex 5 gets involved. Complex 5 has the F0, that is the uh, a piece which is fixed within this membrane. So we call it as the F0 component, so which we call it as integral membrane protein complex, and the F1 headpiece. Okay, F0 component and F1 headpiece. So from here, you know that the protons, they pass through this from the intermembrane space to the matrix to down the electron pump gradient. And as a result of it, the enzyme ATP synthase is activated and there is synthesis of ATP. So the net gain of ATP from each glucose molecule is calculated based on the following assumptions. Okay, so the net gain of ATP from each glucose molecule is calculated based on the following assumptions. All steps in glycolysis, TCA cycle, and ETS occur sequentially and orderly. So first is glycolysis, after glycolysis, the TCA cycle. After the TCA cycle, ETS occurs sequentially and in order. So this is the pattern that we take for the calculation of net gain of ATP. So you can see here. So from the glycolysis, the sequential process and orderly process from glycolysis to the formation of acetyl coenzyme A, then entering into the Krebs cycle, and finally into the electron transport chain. So two ATPs are formed during this glycolysis, and there is Krebs cycle, there is two ATPs formed. Electron transport chain, there is a release of 32 ATP from this FADH2 and NADH2. So 8 NADH2 and 2 FADH2. So 8 into 3, 24. 2 FADH2 is uh, so 2 FADH2. They are going to fetch around 4 of this ATP. Okay. So the NADH, second thing is the NADH synthesized in glycolysis is transferred into mitochondria and undergoes oxidative phosphorylation. So the NADH2 which is synthesized here is transferred into mitochondria and they undergo oxidation. So that will be taken into the electron transport chain. So that is another 2 NADH2. So that will be 4. So 28 plus 4 is 32 ATP. So 2 NADH2 from the glycolysis should also be calculated. So the NADH2 synthesized in glycolysis is transferred into mitochondria and they undergo oxidative phosphorylation. The intermediates in the pathway are not used to synthesize any other compounds. So intermediates in the pathway. So these are all the assumptions for the net gain obtained through during this respiration process. The NADH synthesized in glycolysis is transferred into mitochondria and they undergo oxidative phosphorylation. The intermediates in the pathway are not used to synthesize any other compound. So we also consider this for the calculation of the net gain of ATP. Only glucose is being respired. Other alternative substrates are not entered in the pathway at any stages. So this assumptions we make whenever we are calculating the respiratory balance sheet. So the net gain of ATP from each glucose molecule during respiration is calculated based on the following assumptions. There is an orderly and sequential process. All steps in glycolysis, TCA cycle, and ETS, they occur in sequentially and orderly fashion. The NADH synthesized in glycolysis is transferred into mitochondria. The NADH synthesized in glycolysis is transferred into mitochondria and it undergoes oxidative phosphorylation. So the NADH to 2 NADH2 are synthesized during glycolysis. They are transferred to this oxidative phosphorylation. So the intermediates in the pathway are not used to synthesize any other compounds. So the intermediates that are obtained in the pathway are not used to synthesize any other compounds. Only glucose is being respired. Other alternative substances are not entered in the pathway at any stages. So only glucose is being respired and other alternative substrates are not entered in the pathway at any stages like lipids or amino acids. So they are not entering into the pathway at any other stages. So keeping these assumptions, so we have calculated the total number of ATPs that are going to be there. So such assumptions are not valid, but in reality, does it take place during in our cells? No, such assumptions are not valid because in reality, all pathways work simultaneously and do not take place one after another. So they're all taking place simultaneously. It might be previous cycle or present cycle. So these assumptions are not valid in reality because all pathways work simultaneously. They are all taking place at the same time. ಒಂದೇ ಸಲ ನಡೆಯುತ್ತೆ. Okay, simultaneously means at the same time and do not take place one after another. Like we had said, after glycolysis, there would be the formation of acetyl coenzyme A. After the formation of acetyl coenzyme A, there would be Krebs cycle. After the Krebs cycle, there would be electron transport chain. So this doesn't follow this sort of fashion in reality. So they might be taking place simultaneously and substrates enter the pathway and are not withdrawn from it as and when necessary. ATP is utilized as and when needed. Okay, so they are not like they are all going to be accumulated still they form 36 ATP. That doesn't take place in nature. ATP is utilized as and when needed. So the enzymatic rates are controlled by multiple means. Okay, such calculations are used to whatever calculations we are doing in the respiratory balance sheet. It is used only for the purpose of understanding how many ATPs might be formed from the entire respiration process. But in nature, in reality, it doesn't occur. So such calculations of this respiratory balance sheet are used to appreciate the efficiency of the living system in extraction and storing energy. So that is the only purpose of doing all this respiratory balance sheet. So it is used to appreciate the efficiency of the living system in extraction and also in storing energy. So now the final respiratory balance sheet we have. So during glycolysis, 2 ATP is directly produced and 2 molecules of NADH2 are synthesized during glycolysis. So that is equivalent to 6 ATP. 2 into 3 ATP, that is 6 ATP. And oxidative decarboxylation, so that is the formation of acetyl coenzyme A from pyruvic acid. You know that there is going to be synthesis of 2 molecules of NADH2. So oxidative decarboxylation, there is synthesis of 2 NADH2 and so it is equivalent to 6 ATP. During TCA cycle, there is 6 NADH2 being synthesized for a single glucose molecule. They undergo 2 TCA cycle. So 6 NADH2 is synthesized, which is equivalent to 18 ATP, and 2 FADH2 is synthesized, which is equivalent to 4 ATP, and 2 GTP is synthesized, which is equivalent to 2 ATP. So totally, if you calculate all of them, you're going to get 38 ATP. So 2 ATP molecules are spent for transporting 2 NADH molecules from the uh, cytoplasm to the mitochondria. So they spend around 2 ATP molecules. So from 38 ATP, we have to remove this 2 ATP. The net gain from the respiration of a single glucose molecule is going to be 36 ATP molecules. So the net gain that we are obtaining from a single glucose molecule is 36 ATP molecules. So the here the schematic representation they have given. So during glycolysis, that is glucose forming 2 molecules of pyruvate, there is synthesis of 2 ATP as well as 2 NADH2. So there that is going to be 2 ATP. 2 NADH2 is 2 into 3. So that is going to be around 6 ATP. And during the oxidative carboxylation, where pyruvic acid is converted into acetyl coenzyme A. There is synthesis of 2 NADH2, which is equivalent to 6 ATP. Okay, so the next step is the Krebs cycle. Krebs cycle, there is synthesis of 2 ATP, 6 NADH2, which is equivalent to 18 ATP. Free after this two pyruvic acid molecules, they undergo two Krebs cycle. But a single Krebs cycle is 3 NADH2. That you should be very much aware about. So totally it is 18 ATP, 4 ATP, and total net yield is 36 ATP. So you might ask, sir, here it should be 6 ATP, but for transporting this NADH2 into mitochondria, 2 ATP is spent. So that expenditure of 2 ATP takes place. That is, we have removed it from 2 NADH2. So it has become 4 ATP. Okay. So overall, it is total net ATP yield is 36 ATP molecules from a single glucose molecule during aerobic respiration. Okay. So now let us compare between the fermentation and aerobic respiration. What is fermentation? It is a partial breakdown of glucose. Aerobic respiration, on the other hand, is complete breakdown of glucose into carbon dioxide and water. See, fermentation is partial breakdown of glucose. Aerobic respiration is complete breakdown of glucose to carbon dioxide and water. So the fermentation, so you know there are different types of fermentation, alcoholic fermentation, lactic acid fermentation. The net gain of ATP is going to be only 2 ATP molecules, whereas in aerobic respiration, the net gain is 36 ATP molecules. And fermentation, NADH is oxidized to NAD plus very slowly, whereas in aerobic respiration, NADH is oxidized to NAD plus very vigorously, or very fastly they are oxidized to NAD plus in case of aerobic respiration. So the next is concept is amphibolic pathway. So this cycle, you can see it even in your textbooks, you have this. See, glucose is the favored substrate for respiration. The most favored substrate for respiration is glucose. Okay, so all carbohydrates are first converted into glucose for the respiration process to take place. Other substrates are also respired, like there is fats. Fats break down to form fatty acid and glycerol. Okay, and uh, and fatty acids and glycerol, they break down to the fatty acids are degraded to acetyl coenzyme A, and they enter the pathway. So glycerol is converted to phosphoglyceraldehyde and they enter the pathway. So glycerol is converted to phosphoglyceraldehyde, okay, and they enter into the glycolysis pathway. So this is how the fatty acid and glycerol, they enter. See, fatty acid directly enters into the Krebs cycle uh, as acetyl coenzyme A, and glycerol, they are going to be converted to phosphoglyceraldehyde, and they enter this glycolysis pathway from the phosphoglyceraldehyde stage. So proteins are degraded by proteases into amino acids. So after deamination, so the amino acids, after each amino acid, after deamination, losing the amino group, they enter into the pathway at some stage in the Krebs cycle as pyruvate or acetyl coenzyme A. So the amino acids undergoes deamination and enter the pathway at some stage in the Krebs cycle or as pyruvate or acetyl coenzyme A. They can directly enter into the Krebs cycle or as pyruvate or acetyl coenzyme A. So this is what you notice. So the respiratory pathway is generally considered as a catabolic pathway, but it involves both anabolism synthesis and catabolism breakdown. So it is also called as amphibolic pathway. Since the respiratory pathway is generally considered as a catabolic pathway, but it involves both synthesis, that is anabolism, and catabolism breakdown. So we generally call it as amphibolic pathway. Example, fatty acids break. break down to acetyl coenzyme A before entering the respiratory pathway. So the fatty acids are going to break down into acetyl coenzyme A before entering into respiratory pathway. But when the organisms need to synthesize fatty acids, acetyl coenzyme A withdraw from the respiratory pathway. Similarly, during breakdown and synthesis of protein, respiratory intermediates are involved. So there is breakdown as well as synthesis of protein. The respiratory intermediates are involved. So the respiratory pathway is called as amphibolic pathway because there is not only the breakdown but also synthesis takes place. That is the reason we call it as amphibolic pathway. So during breakdown, fatty acids break down to form acetyl coenzyme A before entering the respiratory pathway. But when the organism needs to synthesize fatty acids, acetyl coenzyme may withdraw from the respiratory pathway. So there is synthesis also. That is the reason here we call it as amphibolic pathway. Similarly, the breakdown and synthesis of protein, the respiratory intermediates are involved. So the respiratory intermediates are involved even in the synthesis of amino acids. That is why we also call this as not only catabolic but also anabolic pathway. Final concept of this chapter is the respiratory quotient or RQ. We also call it as respiratory ratio. So respiratory quotient is the ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed. So respiratory quotient is the ratio of volume of carbon dioxide evolved during respiration to the volume of oxygen consumed. So if it is a carbohydrate, 6 carbon dioxide are liberated, 6 oxygen molecules are consumed. So 6 divided by 6 is 1. So respiratory quotient for carbohydrate is equal to 1. RQ for carbohydrate is 1. See, respiratory quotient is also called as RQ. So it is also called as respiratory ratio. So the respiratory quotient is the volume of carbon dioxide evolved to the volume of oxygen consumed during the respiration process. We call it as respiratory quotient. So the respiratory quotient for carbohydrate equals 1 because they have 6 carbon dioxide are liberated and 6 oxygen is consumed. So 6 divided by 6 equals 1. Now RQ, the respiratory quotient value depends upon the type of respiratory substrate. For respiratory quotient for carbohydrates equals 1 because equal amount of carbon dioxide and oxygen are evolved. So here you can notice this. So 1 glucose molecule plus 6 oxygen, 6 carbon dioxide is liberated, 6 water molecules are liberated, energy is formed. So RQ is 6 carbon dioxide divided by 6 oxygen. So the RQ value for carbohydrate is 1. So the RQ value for carbohydrate is 1. Now similarly, RQ for fats, respiratory quotient for fats is always less than 1. So calculations for a fatty acid like say tripalmitin is shown. So tripalmitin is C to the power of 51 H to the power of 98 O6 2 plus 145 oxygen. So and 102 carbon dioxide plus 98 molecules of water is given. So if you take the carbon dioxide released is 102 carbon dioxide, oxygen consumed is 145. So 102 divided by 145, it is going to be zero point 0.7. So for tripalmitin, so the respiratory quotient for proteins is equal to zero point 0.9. See, respiratory quotient for fats is less than 1. So respiratory quotient for protein is zero point 0.9. In living organism, respiratory substances are often more than 1. So pure proteins or fats are never used as respiratory substrates. So respiratory quotient for fats and proteins are less than 1. They are never used as respiratory substrates. So that is one point that we have to remember about. So the respiratory Substances are often more than one. So pure proteins or fats are never used as respiratory substrates. So the respiratory quotient is greater than one for organic acids. Okay. So these are the things that you have to remember for the respiration chapter. So that completes our discussion on respiration chapter. So have you understood about it, dear students? Any doubts or clarification?